Hi, Miss Nikki here. Welcome to Chapter 28, Pregnancy and Development. I thought I'd open with this beautiful slide. So you can see here, this is an embryo. It's about 7 weeks. You can see the umbilical cord. All of this tissue out here, this is called chorionic villi. This structure right here is called the chorion. And then this structure here is amniotic. Either amniotic sac filled with amniotic fluid or amniotic membrane. So we're going to talk about how the embryo basically creates these structures. We call them extra embryonic structures. And then what is mom contributing? These chorionic villi are sitting in pools of mom's blood. So we'll talk a little bit more about cardiovascular between the mom and the growing embryo when we get to the end of this PowerPoint. Some basic terms before we move on. We have pregnancy. So that's everything from fertilization. So when the egg and the sperm come together and we now have a zygote or a fertilized egg, until the child is born, all of that is labeled as pregnancy. This word conceptus, we don't usually use it that often, but this is the term for developing offspring. Your gestational period is about 120 days, so the time from the last menstrual period until birth. But remember, if this is the last time you menstruated, it takes about 10 days for ovulation, right? So this is our guesstimate. That's why there's not an exact date, right? And then we use these two terms when we're talking about the conceptus, the developing offspring. We either use embryo or we use the term fetus. So basically in the very beginning, it's an embryo through week 8. So when you start week 9, we now start calling it a fetus. So it's a fetus from week 9 until the time that it's born. Here's a nice figure from your textbook. It shows you fertilization through a 12-week-old fetus. And you can see here, very tiny for fertilization. Week 1 conceptus, very small, maybe 1 millimeter. This is a meter stick. The large numbers are actually centimeters, but each of these little tiny hash marks, those are 1 millimeter. So if I'm at a 3 millimeter, 3-week-old embryo, I'm talking 3 of these little hash marks. That's how large a 3-week-old embryo is. And then at 5 weeks, I'm about 10. So 10 millimeters would be from about here to here. So 10 millimeters equals 1 centimeter, just in case anybody wants to know. And then an 8-week-old embryo, we're at about 22. So now we're, here to here. And then here we are at a 3-month-old fetus, and we're at about 90. So if this is 100, we're just a little short of that. So we're probably about there. So that's 3 months. We still have another 6 months to go, right? So I just wanted you to see how teeny tiny this is, and that we can still kind of see the outline of the body plan by this 11 or 12-week mark here. So let's talk about fertilization from the egg to the zygote. So remember, zygote is fertilized egg or ovum, right? Okay, so before fertilization can occur, the sperm must reach the secondary oocyte. And then you're going to have that final division. So make sure that you're paying attention to the fact that meiosis 2 is not completed until after the sperm penetrates the secondary oocyte. The oocyte is viable for about a day, and sperm is viable for about 2 days after ejaculation. So we're talking about 2 days before ovulation and about a day after ovulation, fertilization can occur. So what is fertilization? When the sperm's chromosomes combine with the secondary oocyte and form a fertilized egg, which you're going to call a zygote. When we last spoke about sperm production, we talked about the fact that you could have hundreds of millions of sperm released during ejaculation. So of all of those hundreds of millions of sperm, what can happen to them? Well, some of them are going to get leaked out of the vagina immediately after they're deposited. Some are going to get destroyed. Remember, the vaginal environment is exposed to the external environment, so it's going to be a little bit acidic so that you can't grow bacteria or fungus in the vaginal canal. some of the sperm are going to fail to make it through the cervix. Remember the fornices? I said kind of the little pockets for dumb sperm. So some of the sperm are going to get stuck. They're not going to make it through the cervix. Also remember that cervix has mucus. All right, so some are going to get dispersed into the uterine cavity. Some are going to get destroyed by phagocytes, so specifically macrophages. Again, because the vagina is exposed to the external environment, you're going to have lots of macrophages kind of patrolling and making sure that nothing gets past their defenses, right? And some stuff will still, right? But trying to stop the majority. So we've kind of already talked about this, but I just want to make sure that you realize that only a few thousand are actually going to reach the uterine tube. So this is kind of release as many as you possibly can, you know, hundreds of millions, because you're playing the numbers game. Only a few hundred to a few thousand are actually going to make it to the uterine tube and make it to the ampulla of the uterine tube where the oocyte hopefully is picked up. So we have to talk about capacitation. So basically the sperm have to be capacitated before they can penetrate the oocyte. So we had our flagella, we have our mid piece that has all of our mitochondria, we have the head which has DNA, but then we also have this coating around the head of the sperm, and then in the very front section we have an acrosomal enzyme. Sorry, running out of room. There we go. So the sperm have to be able to move, right? So they're moving their way through the reproductive tract of the female. We have to weaken the cell membrane, right? So this covering, this cell membrane, we have to break it down a little bit because we've got to get these acrosomal enzymes out. The secretions of the female tract are going to weaken the acrosomal membrane. And then the sperm might, this is still in research, right, might have olfactory receptors that can follow the chemical trail by the egg. So depending on, remember you have two ovaries, so you have the ovaries over here, you have your little fimbriae, and then you have your uterine tube leading to your uterus. So it is possible that if this is the one that dropped the oocyte into the uterine tube, that the sperm are probably going to want to go there to meet it, right? Because we're meeting in the ampulla of the uterine tube. Now, again, some people argue infundibulum, but just about that area. So here's the image where we went from early spermatid, and we do all this processing, right? Spermiogenesis, and we end up with our spermatozoa. So here we have the mid piece, which has all the mitochondria. We have the flagella tail. You can see the purple is the DNA. And then you can see this gray material. That's the acrosomal, I'm just going to abbreviate, enzymes. So those acrosomal enzymes are what's going to digest through the outer layer of the oocyte. Because what's the whole point of this? What are we trying to do? We've got our egg, right? We've got our oocyte, and we're trying to deposit the male DNA into the egg so that we can finish meiosis 2, and we can have a zygote with 46 chromosomes if we're talking about a human, right? But you can see there's this outer membrane. So it may be hard, but they're kind of trying to show you with that little, I don't know, beige structure outside of the gray. That is the cell membrane. So that's the cell membrane that was here. We still have a cell membrane, right? If you're a cell, you have to have a cell membrane. So that cell membrane is what has to be eroded away so that this acrosomal enzyme is free so that it can do its digestive duties. So this acrosomal reaction is a little interesting. The book kind of goes into details. There's some things that might confuse you, so I'll try to make it as easy as possible here. The sperm have to breach the oocyte coverings, right? So we've got our oocyte cell membrane, and these are going to have sperm receptors. Then we have our zona pellucida, ELL. And the zona pellucida also has sperm receptors. And then we'd have our oocyte DNA in here. So the sperm have to get through the outer layer, which is the zona pellucida, then they'll fuse with the oocyte cell membrane, and then the DNA will be brought inside where the female DNA is located. So the sperm is going to reach the oocyte. It has to weave its way through the crown of cells, which is the corona radiata. So out here, we also have cells that came with it when it was all ejected from the ovary. So the whole goal is to get the enzymes to be released to digest holes in the zona pellucida. So this enzymes digesting holes in zona pellucida is called the acrosomal reaction. The sperm heads bind to the sperm binding receptors in the zona pellucida first. So this is kind of step one, right? The sperm have to bind the receptors in the zona pellucida. That's going to cause calcium channels to open, and it's this inflow of calcium into the sperm that triggers the acrosomal reaction. So again, you don't want this acrosomal enzyme just doing whatever it wants whenever it wants, right? It could autolyse sperm, right? Could the acrosomal enzyme start digesting sperm? Yeah. Sperm DNA? Yeah. So you want to have an activation for this. So the sperm has to bind the zona pellucida. That'll open up calcium channels, and then that will trigger the sperm to start the acrosomal reaction so that all the acrosomal enzyme is activated and released, and it's basically chewing a hole in the zona pellucida because the sperm have to get to the next layer, which is the oocyte cell membrane, which again has sperm receptors. So to me, this is kind of like a two-step process. We got two barriers. We have a zona pellucida with receptors, and then we also have a cell membrane with receptors. You can see here that the sperm are making their way in, and we kind of blew up this section. So the sperm have to weave their way past the corona radiata to get to the zona pellucida. So this is the zona pellucida. These tiny little spots, those are the sperm binding receptors. So the sperm is going to bind the sperm binding receptor in the zona pellucida first. That's going to cause an influx of calcium into the sperm, and that's going to trigger the acrosomal reaction, get the enzymes released, and the enzymes are going to start chewing through the zona pellucida or digesting the zona pellucida. So when triggered by a calcium influx, enzymes from many sperm are released, and it can digest holes in the zona pellucida. So this is a team effort. Hundreds of acrosomes in the region all release their enzymes at the same time to digest a hole in the zona pellucida. Many sperm are needed to clear the path to the oocyte membrane, but only one sperm is going to get through and deposit its DNA into the oocyte, hopefully. In this image, we can see the acrosomal reaction occurring. So the acrosomal enzymes have been released, they're digesting the zona pellucida, and then finally the sperm makes it to the oocyte membrane. So these are the sperm receptors that are on the oocyte cell membrane. So what's going to happen is the sperm is going to bind the sperm binding receptor on the oocyte cell membrane, and then you're going to have a fusion. So basically the cell membrane of the sperm and the cell membrane of the oocyte are going to fuse so that we can bring in endocytose this DNA. But something else has to happen. Once the DNA gets in, once we have the sperm nucleus inside the egg, we want to stop any other sperm in the process. So let's say that this sperm right here has made it halfway through the zona pellucida. We want to kick that sperm off because we do not want it to get to dropping its DNA off inside the oocyte. So a couple of things happen. These cortical granules that are right on the inside of the oocyte cell membrane are basically going to be activated. So when this fusion occurs, when the sperm cell membrane fuses with the oocyte cell membrane, it triggers these cortical granules to release. They release their contents. They're going to cause calcium levels in the oocyte to rise, And that's going to cause the zona pellucida to become hardened, and that pushes off any sperm that happened to be maybe halfway through the zona pellucida at that point. And you can see here that the sperm receptors are gone. So if directly following this first sperm, if another sperm happened to travel the same exact path, it doesn't have anywhere to fuse. It won't be able to deposit its sperm nucleus inside the oocyte membrane. So these are ways that we block polyspermy. We want monospermy. This is just more detailed words based on the picture that we just saw. So we know that a single sperm is going to make its way towards the oocyte membrane. It's going to bind the oocyte plasma membrane, specifically at the sperm binding receptor, and it's going to cause a fusion of the two membranes. When this fusion happens, it allows for the sperm contents to enter the oocyte. So the tail and the other parts are going to be left behind, hopefully, and only one sperm DNA is allowed to penetrate the oocyte. So this is what we want. We want monospermy. And why? Because the oocyte, we want 23 chromosomes from the oocyte, and we want 23 chromosomes from the sperm, so that when we put them together, we have 46 total, right? If we're human, if we're talking about humans. Let's walk through this slide. It's another kind of view. Just be careful. Don't think it's linear, right? They're just trying to show you this would be step 1, this would be step 2, this would be step 3, this would be 4, right? So they're showing you the steps, but this is not all happening at the same time. So here we have our sperm, and they're weaving their way through the corona radiata. They approach the zona pellucida. In this case, it's kind of this yellowy color. The binding of the sperm to the sperm receptors, here they're a little more specific. They're called ZP3 molecules. So the sperm is going to bind the sperm binding receptors inside the zona pellucida. That's going to cause a rise in calcium inside the sperm, and that's going to trigger the acrosomal reaction. The acrosomal enzymes are going to start digesting the zona pellucida, clearing a path to get to the oocyte plasma membrane. So once the sperm binds the oocyte sperm receptor, you're going to see a fusion of the oocyte plasma membrane and the sperm plasma membrane. The sperm nucleus is going to be deposited inside the oocyte, and then we're going to have this cortical reaction. So these cortical granules are going to release their contents. You're going to have a rise in calcium inside the oocyte. That's going to push off, it's going to harden the zona pellucida and push off any receptors or sperm that would be in the zona pellucida. So the increased calcium hardens the zona pellucida and pushes out any sperm that might be still in the zona pellucida. And you can see that this cortical reaction also destroys any of the sperm receptors that are in the oocyte membrane. So we kind of have a twofold kind of process there, right? Get rid of the sperm receptors on the oocyte plasma membrane, and then harden the zona pellucida to push any excess sperm off. So in humans, we want monospermy. There are two mechanisms that I just explained. This is kind of just the words. So basically, if the sperm binding receptors on the oocyte plasma membrane, if those are all shed, then no other sperm, even if they made it through the zona pellucida, no sperm would be able to bind and get inside of the oocyte or deposit its DNA inside the oocyte. So we call this the fast block polyspermy. The second reaction that happens is called the zona reaction. So when the sperm DNA enters the oocyte, it triggers calcium surge in the oocyte. So this is an increase in calcium in the oocyte, and that's going to do two things. One, it's going to activate the completion of meiosis II. So remember, we're arrested in, I think it's metaphase 2. when the oocyte is ejected from the ovary. So we have to finish that, but we won't finish it until the sperm enter, and we'll come back and talk about that. The second thing that happens is the cortical reaction. So we saw those cortical granules. They start releasing their contents, their enzymes, and those enzymes are going to get rid of or destroy the ZP3 receptors that were in the zona pellucida. So the enzymes that are inside the cortical granules are going to destroy the zona pellucida sperm binding receptors. The egg is going to swell, and we also said it's going to harden, right? And that is going to cause any sperm that might be attached to the zona pellucida to be pushed off. And this is sometimes called the slow block to polyspermy. But basically, we want both of these things to happen, right? We want to push off any sperm that might be in the zona pellucida, and then we also want to get rid of any sperm binding receptors on the oocyte membrane so that we will not get another sperm DNA inserted into the oocyte. Here's the fun part, back to meiosis II. So remember that the oocyte is arrested in meta 2. When the sperm enters the oocyte, that calcium surge triggers the completion of meiosis II. So remember, we're at this part. We've got 46 chromosomes. We have to finish this split. All right, we had our little polar body over here that had 46 chromosomes, right? And then that one may or may not. Sometimes the polar bodies don't split again, sometimes they will. So you've got your first 46 over here. This is the one that you donated all of the cytoplasm to, right, and the nutrients. It's arrested in metaphase 2. We still have to do this split. So the sperm is going to enter the oocyte. It's going to trigger the completion of meiosis II. Then you're going to see the membranes of the two pronuclei rupture and the chromosomes combine. So they call it pronuclei, right? It's not the real nucleus yet. They're going to rupture, then they're going to shuffle all the chromosomes together, and then that'll form the diploid. For us, total 46 chromosomes, right? So fertilization is the moment that the chromosomes combine. Here we have the sperm nucleus. So we went through all the process, got through the zona pellucida, got through the oocyte, plasma membrane, fused membranes, and pushed the sperm nucleus inside the oocyte. Over here on the other side, the sperm entering triggered us to finish meiosis II. Right? We were arrested in metaphase. That's why they're kind of lined up in the middle there. This would be that polar body that had 46 total chromosomes. This would be the other 46 chromosomes. And so once the sperm enters, they go through that cortical reaction, and that triggers meiosis II to complete. So now we should have 3 polar bodies, right? So this is going to split, so that accounts for 1 polar body, 2 polar bodies. What's going to happen to this? It's going to do 23 and 23, and one of those is going to become a polar body. So now we have our female pronucleus. So this has 23 chromosomes. And then we have our 3 polar bodies, and they should all have 23 chromosomes as well. So now the female pronucleus and the male pronucleus are going to fuse. So when the pronucleus, or pronuclei, excuse me, fuse, you're going to see the chromosomes get intermixed. This is when we start saying this is a fertilization is complete. This is when we start saying this is a zygote. How do we get from a zygote to a blastocyst? How do we get to implantation in the uterus? That's where we have to go, right? So we have this fertilized egg. It's in the ampulla of the uterine tube, and it has to make its way to the uterus. The entire time that it's moving, it's also dividing. So embryonic development continues as the embryo travels from the uterine tube to the uterus. That's where it's going to implant. So we have names for these different stages of cell development, right? So from meiotic divisions of the zygote, we're going to go to first cleavage at 36 hours. You're going to have 2 daughter cells, and we call those blastomeres. At 72 hours, we're going to have a morula, which is 16 or more. So remember, we're starting with this zygote. This is 1 cell, and we're copying. So now we have 2 cells, right? And then now we have 4 cells. Don't make me draw all these, right? So we just keep doubling and doubling and doubling. So it starts as a zygote. Then we go to blastomere. Then we're going to go to morula. And then we end up at blastocyst. So I'd make sure I could write that out, right? Zygote, blastomere, morula, blastocyst. So at about day 4, we're at about 100 cells, and this is called a blastocyst. When you see the image coming up, you're going to see that there are certain cells that are called trophoblast cells, and then there are certain cells that are called the inner cell mass. So these trophoblast cells are important because they're going to form those structures that we saw in the very first slide that form the placenta and the chorion. Then we have the inner cell mass, which is actually going to have the 3 embryonic membranes, the mesoderm, the endoderm, right, ectoderm, that are going to create all of the structures. This is a nice visual representation of the division that's happening. It actually goes from ovulation to fertilization to the stages, cell stages, as the zygote is moving to implant in the uterus. So here we have our ovulation, we have our oocyte. It gets picked up by the fembrae, it gets put into the infundibulum of the uterine tube. The sperm meet the egg at the ampulla of the uterine tube, and fertilization occurs. Then as the fertilized egg, or the zygote, right, so at this point we now call it zygote, as the zygote is moving through the uterine tube, it's doing this division. So it's going from a blastomere to a morula, and then by the time it gets here, we can see the blastocyst has different cell types. So we have this outer ring, which are trophoblast cells, and then we have this inner mass, right? So we call it inner mass cell. So this is also why the oocyte had to be large, right? We had to have this one really large oocyte and then these 3 polar bodies. The polar bodies were keeping the DNA away, but they were donating their cytoplasm and nutrients, because we need a lot of nutrients to keep dividing and then to embed in the uterus. So that's why the egg, the oocyte or the ovum, is really large. Implantation. So about 6 to 7 days after ovulation, that's about how long it takes the journey to get from the ampulla of the uterine tube to the endometrium of the uterus, and that's where we're going to implant. The trophoblast cells adhere to the uterine wall and causes this inflammatory response. So the endometrium, we're going to see blood vessels become more permeable. Remember that inflammatory response we called diapedesis? We said the opening of the capillaries, right? So the blood vessels become more permeable. In this image, you can see the endometrium. So all of this is endometrium. We have the uterine epithelial cells, and you'll see that the trophoblasts are on the outside. The trophoblasts are what's going to direct kind of the invagination or the uptake of the embryo into the uterine wall. So you can see there's a blastocyst cavity. This whole thing is called a blastocyst, but you can see there's a blastocyst cavity. All the cells on the outside are trophoblasts, and then we have the inner cell mass here. So basically the trophoblast is going to make all the membranes necessary for embryo survival in this environment, and the inner cell mass is actually going to become the embryo. Can't ever be easy, right? So what's going to happen to these trophoblast cells? They're going to proliferate, they're going to copy, and they're going to form 2 distinct layers. You're going to have cytotrophoblasts, and you're going to have syncytiotrophoblasts. trophoblasts. So you're going to have an inner layer of trophoblasts, right? And you're going to have an outer layer. As the endometrium is eroded, as the blastocyst is burrowing into the lining, you're going to have pools of blood. The endometrial cells will then cover and seal off the implanted blastocyst. This is a nice slide to show you how the trophoblast cells, so remember these were the trophoblasts, they differentiate into these two different types. So the cytotrophoblasts are these cells here. They're still covering that inner cell mass. And then the syncytiotrophoblasts are dividing and expanding into the endometrial lining of the uterus. Remember, this is where we have lots of blood vessels. So what's going to happen is as the syncytiotrophoblasts divide, this embryo is going, at this point it's a blastocyst, is going to be brought into the endometrium. So basically completely burrows into or is engulfed by endometrial cells. If you went to the drugstore and you picked up a pregnancy test, you urinate on the stick and it turns blue or pink, or it has one line or two lines, they're all different, right? What are you measuring? What hormone are you making when you're pregnant that you don't make any other time? Well, that is human chorionic gonadotropin. Human chorionic gonadotropin is only secreted by trophoblast cells. The only way you have trophoblast cells is if you went through fertilization, right? If you have zygote and then you did blastomere and then you did your morula and then eventually you came out with your blastocyte, right? And then the blastocyte differentiates into the trophoblasts and the inner cell mass, right? So what's the only way you have trophoblast cells? Well, you had to have a zygote, right? So the trophoblast cells secrete the HCG, and HCG does a number of things. One, it's going to prompt the corpus luteum to continue secreting progesterone and estrogen. This is going to stop menstruation. It's going to promote placental development. And it's going to rise until the end of the second month. So about 8 weeks. And then it's going to decline because the placenta begins to secrete enough estrogen and progesterone to maintain the pregnancy. So this slide is important for you to realize that the corpus luteum must be maintained in order to secrete estrogen and progesterone until the placenta is fully formed and can secrete its own estrogen and progesterone to maintain the pregnancy. And what's the hormone that signals that? It's going to be human chorionic gonadotropin. And that's the hormone that we use if we're trying to test whether somebody is pregnant or not. This is a nice visual representation. So we can see at the beginning of ovulation and fertilization, there's not a lot of estrogen and progesterone. We have a little, right? It's starting to rise. So this estrogen and progesterone is specifically because of the corpus luteum. And what hormone is directing the corpus luteum to continue to release estrogen and progesterone? That's the human gonadotropin. And we can see that it peaks at about week 8. And then it starts to decline. Estrogen and progesterone really pick up through, let's say, week 15-ish out to birth. And this is as the placenta is growing, right, and has enough cells to maintain the estrogen and progesterone level. So this is why the corpus luteum is really important in the beginning phases. If something happens to the corpus luteum, you're not going to be able to maintain pregnancy. Throughout the chapter, hopefully you were thinking about all of the possible breakdowns in this system, right? What could go wrong that would not allow pregnancy to occur? So one of the things to think about is failed implantation. So just because you have fertilization, just because you have a zygote, doesn't guarantee you that that zygote is going to continue all of its division to a blastocyst, blastocyst, blastocyte, and it may not implant into the uterus, right? So about 2/3 of all zygotes formed fail to implant by the first week or spontaneously abort. That's a lot. People don't think about the fact that we have probably self-aborted many zygotes, either from too many chromosomes or from failure to implant, right? So 30% of implanted embryos later miscarry because of some genetic defect, especially as the chromosomes divide and we start going through organ development. So just something to think about, that it's all of these steps need to occur, and they need to occur perfectly in order for us to carry a child to full term. Now we need to talk about the formation of the placenta, placentation. So basically this is the embryonic tissues and the maternal tissues. So you can see here, number one, embryonic tissues, we're going to talk about the chorion and the chorionic villi. And then two here, we're going to talk about the maternal tissue, which is really just blood-filled lacunae. Remember, that meant lakes. So remember how the blastocyst is burrowing into the endometrial lining, it's also breaking down maternal endometrial tissue and developing these pools of blood. Here's a nice figure to kind of go along with the last slide. We can see that the blastocyst is burrowing into the endometrial lining. We can see these maternal blood vessels that are going to kind of start filling in as the syncytial trophoblasts proliferate out. And if you remember, we had that cytotrophoblast cell ring. Those cytotrophoblasts are going to start forming the chorion. And then after that, what's going to happen is the chorion is going to start forming these chorionic villi. They're like capillaries, right? And so the chorionic villi are sitting in pools of mom's blood. Here's another image for you, about 4.5-week-old embryo. So I want to point out the amnion. So that's the amnion, and there's going to be amniotic fluid in here. And then we're going to have the chorion, goes all the way around, and the chorion is where we have these blood vessels coming off, right? And these are called the chorionic villi. Villus for one, villi for multiple. I want you to notice these words, decidua basalis and decidua capillaris. Basically, it's kind of like the front side and the back side. So this is kind of facing, in this case, the embryo. And this is on the posterior or back side of the embryo. And what's going to happen eventually as the embryo grows into a fetus, you're going to have kind of pressure and pushing on this decidua capillaris. And so it's going to become very thin. They're all kind of going to get stacked and squished together as the baby grows. And then eventually you're only going to be able to see kind of like a disc formation. And that disc formation is the decidua basalis. And that is basically what we call the placenta. So if you see kind of this nice little disc shape, that's going to be the decidua basalis. That's the only part that you can see once the embryo/fetus gets to a certain size. And that's where you're going to see the blood vessels. These kind of start shrinking and or get pushed together. I think this is a nice statement just to make sure that we understand the importance of the placenta. So the placenta provides nutrition, right? So food exchange between mom and the growing embryo, respiratory gas exchange, right? So oxygen, carbon dioxide, excretory, so getting rid of waste that the fetus is creating from doing cell division, and endocrine functions. So hormones can be transferred to mom to help direct fetal development. So the placenta here, the key is it's not fully formed and functional until about month 3. So just go back and look over, remember what's providing hormone function, right? Corpus luteum is providing estrogen and progesterone. And then we also have the HCG, which is directing the placental development. So we do have other hormones at play here, but realize that the placenta is not fully formed until month 3. The other thing I want to point out is the book uses this term intermix. So I don't want you to think of maternal capillary beds intertwined with embryonic capillary beds, because that's not what's happening, right? So we have that chorion, and then we have the chorionic villi, and the chorionic villi are sitting in pools of mom's blood. So this is basically trying to point out that there's no interconnections between blood vessels. It's just diffusion happening with the chorionic villi laying in pools of mom's blood. So if the placental hormones are inadequate for any reason, after that 3-month period, the pregnancy is going to be aborted. So throughout pregnancy, you're going to see blood levels increase of estrogens and progesterones, and that's going to help us kind of prepare for not only birth, but also for development of mammary glands for lactation. So just to drive the point home, we can see the maternal arteries here. You can see that the arteries are dropping off into these pools of mom's blood. And then you can see the chorionic villi and how they have access to the pools of mom's blood. So this is really just diffusion, passive transport of waste and nutrients and gases between mother and fetus. You can see here, this is another good example to show you how that decidua capillaris kind of gets squished as the embryo is growing into a fetus, and that you kind of end up with this disc. It's really the decidua basalis, but we're going to go ahead and call it the placenta. So it's the chorionic villi laying in pools of mom's blood. That's truly what the placenta is. Now we're moving on through embryonic development from gastrula to fetus. So gastrula, we're still talking about embryo, right? We're moving from embryo to fetus. Just back up for a second and make sure that you understand all these terms. So zygote was the fertilized egg. Then we moved into a blastomere. Then we moved into a morula. Then we moved into a blastocyst. Now we're moving on to gastrula. So these extra embryonic membranes, these are membranes made by the embryo, right? So the amnion, the chorion, the yolk sac, the allantois. And there are 3 germ layers that are going to start to become present during this gastrulation phase. So we're going to see an ectoderm. So I think of this as outer, mesoderm as middle, and endoderm as inner. And we'll look at which structures are made from these different germ layers. So the first embryonic membrane that I want to talk about is going to develop between the 2 and 3 weeks of development, and this is the amnion. So the amnion is going to be filled with amniotic fluid. It's going to be the closest membrane to the embryo. It's going to provide a buoyant environment. So think of free floating, right? It's going to protect the embryo against any sort of trauma. It's going to help maintain a constant temperature. It's going to allow for the freedom of movement. We're going to see cells that have to fuse together during this point in time, and so they have to have the environment to do that. So something just to pay attention to here, in the beginning, the amniotic fluid comes from the maternal blood. Remember, blood consists of water. So at first, the amniotic fluid is going to come from maternal blood, and then later it's going to come from fetal urine. But remember, this is not the same type of urine that would be excreted from our bodies, right? It's a very watered down version. The fetus is not exposed to, regular food in the environment. They're still getting all of their nutrition from mom in the uterus. So the second extra embryonic membrane that we're going to discuss is the yolk sac. So the yolk sac hangs from the ventral surface of the embryo, and it's going to form the digestive tube. If you remember way back in the very beginning, chapter 1, we talked about mouth to anus, right? We might have talked about alimentary canal, but basically you have a tube that runs all the way from your mouth, right? Pharynx, esophagus, stomach, small intestine, large intestine, anal canal, anus, right? So this digestive tube is going to start to form on the outside. So if we had our embryo here, you're going to see a yolk sac developing on the outside, and that's going to become the digestive tube, which then has to become an internal structure. So there are some children that are born and their digestive system is on the outside of their abdominal cavity. It was never directed to be engulfed into the embryo or fetus. This is also the earliest blood cells are formed in this yolk sac, and then also the cells that end up making gonads. This is where they start. The third and fourth extraembryonic membranes, the allantois, is this small outpocketing that's at the end of the yolk sac. This is the structural basis for the umbilical cord. It also is going to become part of the urinary bladder. So some of these outside structures are eventually going to be brought inside the developing fetus or embryo. The chorion, we've already talked about, helps create the placenta. So the chorion is going to create the chorionic villi, and those chorionic villi are going to be laying in pools of mom's blood, basically setting up what we know as the placenta. So you can see here the yolk sac, right? It's on the ventral side. You can see on the caudal side, you're going to have the vessels of the umbilical cord. That would be where you'd see the allantois. You can see the chorionic villi and the chorion laying in pools of mom's blood. You can see the amnion and the amniotic fluid, which is the closest extraembryonic structure to the growing embryo in this case. Now we're talking about gastrulation, these germ layer formations. So how do we create the ectoderm, the mesoderm, and the endoderm? So at about week 3, we're going to start seeing this embryo that has this 3-layered structure. The first thing you're going to see is something called a primitive streak, and this helps set up the axis of the embryo. I'll show you on the next slide. So this is what I mean. You can see we go from this bi-layered structure. Eventually, we're going to have this tri-layered structure. So when this primitive streak forms, it basically sets up, like they said, the longitudinal boundaries. So this is going to be the head end, and this is going to be the tail end. And we still just have 2 layers here. And then eventually what happens is cells are going to enter this primitive streak, and they're going to start filling in kind of this bottom section. This is going to be the endoderm. And then you're going to have more cells that enter this primitive streak, and they're going to fill in the middle. So this is where we get ecto, endo, and meso. So the ecto were there, and the endo were there. More cells started coming in and filling in this endo, and then the second round of cells started filling in between both the ecto and the endo. And we call that the mesoderm layer. Basically, this is the wording to the slide that we just looked at. So the first cells that enter are going to form the endoderm. The cells that follow next are going to be the mesoderm. They're basically going to push their way between the ectoderm and the endoderm. So those cells are going to come in through the groove created by the primitive streak. Then we're going to see a structure called the notochord. So basically, it's mesodermal cells that serve as axial support. So when I see notochord, I start thinking of axial skeleton. So this should make us think of brain and spinal cord. This image, we can see tail and head. There'd be our primitive streak, which we created our groove, primitive groove. And then we know that cells entered that groove, and we ended up with this tri-layer system. So we have our ecto at the bottom, we have our meso in the middle, and then we have our ecto on the outer. Here's another image of that tri-layer. So we have our endo, and we have our meso in red, and then we have our ectoderm in blue. And so what's going to happen is you're going to see the digestive tube formed. And this is going to also give you an indication of what type of tissues are probably made from each of these. I'll go through the list in just a second, but do you remember how the inner lining of your digestive system is epithelial cells, right? Those that are easily shed off, stratified, non-keratinized, stratified squamous epithelium. Well, they're created probably, if you had to guess, they're created by this endoderm. So basically, each of these germ layers are going to be responsible for turning into or differentiating into a specific type of tissue. Maybe this slide will help kind of put everything into perspective. This actually happens during organogenesis, which is a slide, Organogenesis. There we go. There's a slide coming up where we're going to talk about we move out of the gastrulation phase and then we move into the organogenesis. But I mentioned notochord before, so I wanted you to see that this notochord forms and then this neural tube. We're basically setting up the structure for our spine. So we're setting up our brain and our spinal cord structure here with the neural tube and the notochord. And you can see the yolk sac here. So that other image, what's going to happen to the yolk sac? It's going to be brought into internally and create your digestive tube. So on to the good stuff. Can you tell me what each layer or how each layer is contributing to the growing embryo? So ectoderm, outer, is going to be the nervous system, the skin, the epidermis. If it helps you for nervous system, remember nervous system is dorsal, so maybe that helps think of outer, right? Endo is inner. So these cells are going to become the epithelial linings of the digestive, which we showed you. That was the yellow lining that I showed you from the yolk sac was kind of invaginating into the growing embryo. It's also going to form your respiratory pathway and your urogenital system. So think of anything that has epithelial linings, right? So any tube system, any internal structure that has epithelial linings, it was created from endoderm. And then mesoderm is that catch-all. It's everything else. So usually endoderm and ectoderm are considered epithelium. Remember, skin has epithelium, right? So let's look at a more detailed image from the textbook. Here we have ectoderm, then that lists all the structures, the epidermis, the hair, the glands, the brain, the spinal cord, right? Then we do endoderm, all the epithelial linings and glands, digestive, respiratory tract. You can see why we call mesoderm the catch-all, right? It's everything else. So vertebral discs, right? Ribs. So I'm thinking skeleton, yeah? Myotomes, probably something to do with muscle. How do I know? Because it's got myo, right? Organs, kidneys, gonads, your serous layers, part of your dermis. Remember, this is connective tissue. So bones, joints, ligaments, blood vessels, heart, visceral serous. This one was parietal. The walls of the digestive and respiratory tract. Remember, those all have smooth muscle. So basically, if you can remember that ectoderm is your epidermis and your brain and spinal cord, and your endoderm is any epithelial lining of a tube system within the body, everything else is made from the mesoderm.